antibiotic treatment, drug resistance, and even in the field of ecology. We are also glad to announce the upcoming hackathon on microbiome in health and wellness. This is an initiative to provide students with a platform to showcase their innovative mindset of problem solving. Here, each student will have the opportunity to develop a mini project around the hackathon topic, form a team with at least 2 students, and compete against each other for mentorship, support from established companies, and long-term interest from the business community. The applications of hackathon are in the fields of obesity and precision nutrition, innovation and beauty products, and in pregnancy. So I would like to urge all the participants to be a part of this hackathon and make the most out of such a wonderful opportunity. To know more about the event, you can visit the link that will be provided to you in the chat box by one of our team members. So before we begin with today's session, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. The Omics Logic training has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. And this training has been completed by participants in 6 different specialization tracks, which includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. So how do Omics Logic resources help advance bioinformatics teaching? Omics Logic is a portal for practical and theoretical learning of bioinformatics. It takes a combination of training modules, data analysis tools, curated project datasets, and interactive session with mentors to give the student a clear path up the Bloom's taxonomy pyramid. And no matter what the user's initial experience with bioinformatics is, Omics Logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, and introduction to basic coding languages like R and Python, or even beginning a data analysis project. The program comes with an access to the Omics Logic courses, which includes courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, epigenomics, data science that includes bioinformatics using R and Python, as well as cheminformatics. Users will be given directions to and example projects of different fields, which you could learn from and replicate for your research work. Users will be given directions to access the tBioinfo platform for bioinformatics processing and analysis of data. And the platform includes demo pipeline as well as data management and cloud analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis are performed in different sections on this multi-omics platform. So first of all, let's begin by signing up on the Omics Logic Learn portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features you need to keep in mind. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles, which could be your Facebook, LinkedIn, Instagram, OrcID, or even Twitter, and a brief bio about yourself that will consist of your educational background and research interests. The mentors can view your progress under the Activity tab. The courses you are currently completing will appear under the Courses tab. Finally, after the completion of your course, the certificates will appear under the Certificates tab. So with that, let us begin by signing up on the portal. I'm sharing the link in the chat box. Please click on the link and I'll walk you through the process behind signing up on the portal. All right. Okay, Ojasvi has already shared the link in the chat box. So for users who do not have an existing account on the portal, please click on the Create an Account option that you see here. And here you will be asked to enter your name, email ID, and provide a new password. You also have the option to sign up using any of your social media accounts. This could be your Google account, Facebook account, Apple account, GitHub account, or even Twitter account. And with that, I'll pause for a couple of minutes for the participants to sign up. And once you have signed up, please put a yes in the chat box. Also, if you're facing any technical issues, you can put them in the chat as well, and our team will be happy to help you. So I'm waiting for the responses from the 59 participants that we have. So once you sign up on the portal, please put a yes in the chat box so that I know I can proceed further. All right. Okay, great. So since I already have an account on the portal, I'm going to click on the login now option and click on the sign in option to log in. Once you sign in, you will be able to view the various courses and example projects that are appearing under the courses tab. So here, since most of the participants are interested in metagenomics, when you type the keyword metagenomics, you're able to see the coursework on metagenomics, also the various example projects that include role of high-fat diet on obesity, effect of dietary fiber intake on microbiome, effect of non-antibiotics, and Rett syndrome. And coming to the most important part, as I discussed, when you click on the tab that says welcome with your email ID, it will take you to the account settings. Here, so how do you edit the features that I mentioned before? For that, when you click on the icon that says profile, you'll be able to edit all the details, necessary details to update your account. So this could be your bio. Here you should update your education background and research interests. Name should be in appropriate capitalization, link to any of your social media handles. So in my case, I've linked my LinkedIn account and my org ID. And under the activity tab, the community manager and the mentor will be able to track your learning progress. Once you start completing any courses, it will appear under the courses tab. So in my case, I'm completing the Python course too. And once you have completed any project and uploaded on the portal, it will appear under the projects tab. And if you're enrolling for any of the programs, such as the Metagenomic Data Analysis program, it will appear under the programs tab. And finally, after completing the courses, you'll be able to receive the certificates under the certificates tab. So far, if anyone has any queries, please put them in the chat box. Otherwise, now I'd like to invite Dr. Raghav to start with today's session. So does anyone have any queries so far? If no, I'd like to pass on the stage. Over to you, sir.
Thank you, Sri Gauri. Thank you very much. So let me find ways to share my screen and set up the presentation. Can you see my screen? And
yes, sir.
You can also hear me?
Yes, sir.
Okay, thank you very much. Let me organize this a little bit so that I can follow your chat also. Okay. So welcome to Omics Logic Metagenomics program and session 1. So in this specific course, we are going to introduce you to the bioinformatics analysis of metagenomic sequence data. So in today's session, in this first session, we will start by speaking about genomic sequencing technologies as one of the methods to study microbes and to map out how such data can be used to understand the biology of the host system. Then we will introduce you to several data analysis tools for the metagenomic studies and see a couple of examples where such analysis approaches have been used. So let's start reviewing a little bit of history of microbiology, where this study of microorganisms led to the creation of tools and creation of approaches that are still being used today to continue and to understand the world which we do not see, but yet has such a profound effect on everything around us. So for many years, the smallest things that humans could see was about as wide as a human hair. when the microscope was invented, suddenly we saw a new world that is living under our own eyes, or in our water, in our food, all around us. The first observation of a bacterium was made around in 1673 by Anton van Leeuwenhoek. That's a name that I do not know how to pronounce even after so many years. So thanks to the development in microscopes, and we can now see what he saw 50 to 300 times enlarged. So as the resolution of the tools that we use to observe these microbes and bacteria improved, more and more representatives of these unseen and unknown world have been discovered. Today, the microscopes, as we know, are tremendously powerful, and this allows us to see smaller and smaller and smaller elements that reveal how the world of microorganisms function and how they are related to the other microorganisms and to our own host's environment. So in this particular image, we can see a super high resolution picture of bacteriophages that are in green attacking E. coli bacteria. So this is imaged with the helium-ion technology, which produces the super high resolution picture of this such a small world. So the field microbiology is defined not only by the organisms that we study, but also by the tools that is used to study them. And the tools that we use have evolved to allow us to study and categorize these microorganisms, right? The technology for example, for understanding bacteria is usually in bacterial culturing, coupled with techniques to stain this bacteria. And these methods improves our ability to isolate, to classify, and to determine whether a bacteria or a microbe is a cause of human disease, or it is even appropriate to select some of the specific treatments attacking and neutralizing those specific bacteria, right? For example, the discovery of culture media allowed for the development of this microbiology in around 18th and 19th century. Bacterial culture is the artificial medium that allows the growth and isolation of bacteria. The first to have isolated bacteria is a scientist that we all very well know, this Louis Pasteur, right? And staining, as we already spoke about, is a technique that is used to differentiate bacterial species into two large groups, that is gram-positive and gram-negative, based on the physical properties of the cell wall of the bacteria. So why is this necessary? So gram staining yields results much more quickly than culturing. And this is extremely important. For example, when an infection would make an important difference in the identifying an infection would make very important differences in the patient's treatment and prognosis. Some of the examples are meningitis and septic arthritis. Each one of these diseases are caused by gram-negative and gram-positive bacteria. So identifying which one treats what is very important, which one causes what is very important to for developing prognosis and better diagnosis. So generally the connection between microorganisms and the diseases that it can cause has been established in 1800s, right? As early as 1800s. And these studies, there are numerous studies that proves or provides important information about the function of the bacteria in isolation. However, more and more studies reveal that these bacteria and the microorganisms, they do not live in isolation, as we used to study them using culture media and using staining methods. But these microorganisms, they live in complex communities, and many members of these complex communities are not known, or not even studied, or completely unknown, and cannot be isolated to study in the culture or any staining media. So today we know that these community of microorganisms, as we call them as microbiota, that make up, for example, human microbiome, is not just unicellular organisms that are living aside each other, but they are highly regulated, highly structural and functionally organized communities of bacteria, virus, and fungi attached to the surfaces of our organs and our tissues, which usually we call them or term them as biofilms, with collaborations among themselves that contribute to their stability and promote their beneficial promote their benefits to our host environment, to the host environment, that is us. So how can we study these multitude of microorganisms that cannot be cultured or that cannot be identified using traditional methods like staining or culturing, etc.? So around 1980s or 1970s, we identified that these microorganisms that are yet not been understood and studied can be explored using the advancements in sequencing technologies and other omics technologies. So due to these advancements in metagenomic sequencing and several other collaborative omics technologies, enormous databases of sequencing data from such microbial communities are available. And this availability of so much of data has led to the emergence of computational metagenomics, which can help us study 99% of the uncultivable microorganisms in context to their natural environment. So the emergence of such metagenomic sequencing methods and generally sequencing methods has opened up a new door in understanding microbiology, and not only in isolation apart from their or away from their natural environment. We can study them in their natural environment and as their functional importance to maintain the balance and symbiosis of the natural environment in a healthy state. So one such example is taking our skin cells or taking our skin samples. So we already spoke about bacteria or microbes that are able to grow in complex communities, and they can actually grow or get adhered to almost all surfaces or every surfaces. And these adherence to every surfaces, and due to these adherence to every surfaces, they form complex communities, and these complex communities of bacteria on any such surfaces or any such living surfaces are termed as biofilms, right? The stability of these communities is probably very important, and is fascinating also to understand from common skin conditions point of view, for example, as eczema, acne, and dry skin in compared to healthy skin. Why? Because forming such distinct communities, these microbes and microorganisms are interconnected, and they are determined to maintain their community, and their balance and their healthy maintenance of their community is very important to us to maintain the healthy state of our skin in this example, right? So any bacterial community or any bacterial group within a biofilm can communicate with each other by producing, by detecting, and by responding to small signal molecules. And this method is called quorum sensing, right? And this such communication benefits the bacterial community for colonization, for defense against competitors, and to adapt themselves to any changes and any stress to the environment, right? And these sensing activities are also involved in the pathogenic potentials of some bacteria and the virulence of some viruses and bacteria, and is therefore extremely important to understand how bacterial infections are controlling and how bacterial infections are affecting the symbiosis or balance of microbial communities and their abundance in these biofilms, right? And inside each community, as we already spoke about, there is generally a balance of the amount of bacteria, the amount of fungi, and the amount of viruses that controls species abundance and species activity. So this identifying this balance of these different species is key to understand the healthy state of the sample. For example, in this case, healthy state of the human skin, or a diseased state of the skin under conditions like eczema, skin, acne, etc., etc. So human microbiota is, for example, estimated to be around 10 to the power 13 or 10 to the power 14 of microbial cells, and that is approximately 1:1 microbial cells to human cells ratio. This number or these figures are derived from the total bacterial cells that is identified in colon, in the organ colon, where we have around 4 into 10 to the power 13 bacteria identified. Colon is the organ, as we now know, harbors the densest number of microbes or microbial communities. However, each one of these microorganisms that is present in environment like colon or any other organ can be explored for, they carry important functional properties and important functional contribution to maintaining the healthiness of that specific tissue and organ, right? And the importance of these specific bacterial community and their functional properties can be explored by characterizing the genomic information of these specific microbiota. And these properties and the genomic information about the microorganisms have been of growing interest to the scientific community. As we can see, it has resulted over 80,000 publications until now, right? And the majority of the studies that we can observe have been focused on understanding the intestinal microbiome, or otherwise called as gut microbiome. But as we discussed, other organs and their microbiota are also becoming increasing topics of interest in the latest metagenomics research trend. For example, inflammatory and microbial influences between gut and between skin for a healthy state and dysbiotic state can be explored, and this is actually explored in this specific publication. And what do I mean by understanding that? And they have actually listed out or explored or tabulated specific strains and specific communities of bacteria that is associated with skin microbiota under variety of disease conditions and under variety of imbalance or dysbiotic skin microbiota conditions, right? In addition to exploring the skin microbiota for the variety of these diseases, one can also relate to the abundance of microbiota that is present in the gut and their influence on the diseases that one observes in the skin. And this relationship is known, or is actually termed as gut-skin axis, right? And for example, how this is related, the intestinal and the epidermal barriers are connected through the circulatory system, as we know, and are visualized here in a very small visualization, in a simplistic visualization manner here, right? The dysbiotic state is characterized, for example, that can be characterized by, for example, impaired or imbalance in the gut microbiome, and it can reduce the mucus layer, it can reduce the immunoglobulin secretion, it can, for example, disrupt the barrier between the microbes and the intestine. It can also, for example, lead to increased intestinal permeation into the bloodstream, and it can also lead to some inflammation that can be observed around the gut. And again, at the skin level, it can lead to impaired skin barrier, imbalance in the skin microbiome, or reduced, for example, antimicrobial peptides production, as we saw in column sensing, right? That can increase the probability of getting skin rashes or lesions, and etc., etc. The gut and the skin dysbiosis or imbalance, right, are connected through the immune imbalance themselves. In this specific example, this publication deals with understanding the imbalance in the production of helper T cells or Th2 cells or T lymphocytes that is skewed when there is no, or when there is when the crosstalk between the gut microbiome and the skin microbiome is disrupted. So if you want to check this publication, I can give you the link for this. This is an exciting and excellent study that relates to microbial community and their effect in skin conditions as such. So coming back to the presentation, such studies, and since 1990s, many microbiome studies have significantly advanced through major initiatives such as metagenomics of the human intestinal tract and the subsequent human microbiome project, and tremendous studies and huge studies have been made possible through several advancements in metagenomic sequencing. And the result of these advancements is that we study not only the gut microbiome, we explore a lot of skin microbiome, lung microbiome, intestinal that we already spoke about, and even brain microbiota are starting to get characterized. Each new project represents vast opportunities and challenges to determine which microorganisms and how their abundance play key role in the development of certain environmental conditions that are necessary for maintaining the healthy functions of human organs, and which abundance level can cause harmful effects to or harmful results to these human organs in general. So in the past, over the past decade, over almost $2 billion has been spent on human microbiome research, and major projects are underway in the United States, in the European Union, China, Canada, France, Ireland, Korea, Japan, etc., etc. And these investments have confirmed the importance of microbiome to human health and development. And for example, to give you a scale, something around 20% of human investment in such research has gone into human microbiome project, which is creating the research resources or the data that is necessary to understand human microbiome, and which is one of the main goals of human microbiome project. That is to create reference data sets and necessary analysis methods, necessary computational techniques, and necessary clinical protocols to understand these reference data sets of healthy American human beings. Right? So it is because of such huge investments and such potential that we know and we also find that several endogenous human microbial communities can contribute to critical metabolic and physiological and immunological characterization of our human body. And this can be done by understanding the differentiation and maturation of the immune system. This can be done by, for example, understanding the microbial influence on microbial communities, influence on the food digestion, on the energy generation, on nutritional levels, metabolic regulation, and control of the fat storage, etc., right? And we can also extend this association to processing and detoxification of the stress and environmental chemicals. We already spoke about the maintenance of skin, and this is also involved in maintenance of the mucosa barrier function, and this is again very much related. The microbial function and their abundance level and their balance is very much related to the development and regulation of pro and anti-inflammatory processes, and they can also be associated with the prevention of invasion and growth of disease-promoting microorganisms, as those pathogenic microorganisms can cause disruption to the microbial community themselves. So what is in store for bioinformaticians as such, right? Metagenomics, as we know, is the genomics analysis of microbial DNA obtained from their natural environment. And the tools that are available to study this metagenomics enable us to analyze or perform population analysis of these unculturable or these previously unknown microbes, and this ability to identify these microbes without any a priori knowledge and actually helps us to open new doors in disciplines like virology, microbiology, ecology, and environmental sciences and biomedicals in research in general. This sequencing-based examination of metagenome has become an extremely powerful tool for generating novel hypotheses, and it will not be an exaggeration to say that it has evolved itself into the primary tool for understanding the microbial community and microbes inside in their natural environment as such. My animation is running slow. Let it run. So what is the difference between metagenomics and other omics studies, right? Unlike other omics studies, metagenomics does not involve or does not study a single organism, but it studies the whole community at once. It can be paraphrased as a snapshot of a collection of organisms directly in their natural environment. And since the genomes that we can sequence are so diverse, this is typically accomplished by sequencing a specific portion of the genome instead of all the genomic material that is given or present in any given sample. And many of these methods are actually, as we know, are actually performed using the next generation sequencing techniques, which either focuses on sequencing the whole genome or the whole transcriptome of the microbial population, as we spoke about. That is one possible option. Or we can also sequence a selected gene of the of the specific gene of a microbe, and this gene is usually the 16S rRNA gene. Or we can also go for studying selected portions of this specific gene, that is 16S or 18S rRNA gene, to study the hypervariable regions that is present in these genes. So when you perform this analysis or when you perform these sequencing methods to sequence the whole genome or transcriptome, you have the flexibility to analyze these sequences and relate to their functional characterization of the microbes. And when you perform 16S rRNA sequencing or selected portion or hypervariable amplicon sequencing, then we have the ability to estimate the abundance levels of these microbes and relate that to the biological conditions of the sample, either it is from the diseased state or healthy state in general. Right? This 16S amplicon sequencing, as we mentioned, is fairly a recently developed technique that is very widely used to study microbiome abundance directly in its environment. Right? This method focuses on isolating 16S or 18S ribosomal RNA and amplifying using PCR amplification methods. Right? This gene, 16S rRNA gene, contains hypervariable regions, which is also it is termed as hypervariable regions because they provide or they contain species-specific signature sequences that is generally used for identifying the species or taxonomic rank of any microbial entity. The benefit of this amplification is actually tied to the nature of sequencing read length, which can actually maximize the effective output from the affordable sequencing technology. Or in other words, since we are not sequencing the whole genome of the bacteria or of the microbes, we are only focusing on specific portion of the genome, we can scale this up to multiple levels at an affordable sequencing rate. So general differences between what one can achieve from shotgun or metagenomics or metatranscriptomic sequencing or amplicon based sequencing is can be summarized as this. As we already spoke about this, the metagenomics or metatranscriptomic sequencing can give you gene prediction information, functional annotation of the genes that are predicted, and it can give you assembly of these genome for microbes that are unknown or that are not characterized. So these information or these analysis method can be achieved using the metagenomics or metatranscriptomics sequencing method, while the amplicon sequencing method or 16S rRNA gene sequencing method can actually give you can give you the abundance level of different microbial population for the different sample sizes that we or for different sample conditions that we have, and relating the abundances and from the abundance level, understanding the balance of different microbial community and different phyla or genus or family of microbial community can be extremely important analysis to understand the biosis or balanced state of these microbial communities in their natural environment. So if we take these take the abundance entity or abundance level of microbial community for a diseased state and compare it with a healthy state, so we are bound to see differences in specific set of or specific genus or phyla of bacteria that can be related to generally can be related to be the marker bacteria or marker species for those specific diseased state in general. Yeah, this is what is it is. So this 16S gene, as we spoke about, are the hypervariable regions that we can sequence from the 16S gene, are the ones that are used to find a specific bacterial taxonomy, or are the ones that are used to identify these bacteria, as we will discuss in a couple of slides. So these genes are generally not identical, right? I mean, the 16S RNA gene and their hypervariable regions, even though they are sequence specific, species specific, they are not identical, but they have their innate diversity and variation, and they are usually combined or clustered into what we call as operational taxonomic unit, which can be used to classify groups of closely related individuals. So these OTUs are actually formed, are actually identified by clustering the genes or the sequences that are that represents either 99% or 97% at identity level. One of the shortcomings of such method is that we may end up pooling two or more than two different species or genus of closely related bacteria together. And in other words, we can even sometimes end up separating bacterial species or bacterial genus that may exhibit a lower identity level, lower sequence identity level at this OTU, at this 97 or 99% threshold. So that is an associated risk, or that is an associated limitation of clustering based on OTUs. But there is another method that is actually developing. That is right now it is actually fully developed, and many of the quantification microbial community quantification methods actually started adopting to amplicon sequence variants, right? So we spoke about the most common type of clustering method for identifying these microbial community has been the operational taxonomic unit, which generally uses a fixed sequence identity cutoff, and that is usually 97% for the sequence from the same species. But however, as we already discussed, different regions vary in and different species vary in their sequence identity range, and sometimes phylogenetically related taxa can share less predictable sequence identity range, and to accommodate them, we cannot reduce the level of sequence identity. So these are all current limitations of these OTU methods. And in recent years, we have come to understand, or we have come to use or introduce denoising through several different algorithms as an alternative to the clustering method that is generally employed to identify or cluster these related sequence together. So these these denoising actually helps us to eliminate sequential errors before forming these clusters, and are hence referred to as amplicon sequence variants. So these ASV or amplicon sequence variants based clustering is technically based on sequence probability rather than sequence identity, as in OTU. The denoising approaches uses very well-established statistical methods to determine, for example, which low prevalence sequence appear more than that would be expected for at a noise level, and therefore that are observed to be valid sequence variants. And they also identify which, for example, sequences are identified or can be identified as noise sequences, and that will be eliminated from there. Further clustering, level isolation, and this results in fewer reads or fewer sequences to proceed to the clustering stage, but it results in consistent custom clusters or consistent sequences that can be identified or that can be related at specific identity level, which is also performed at 97%. So in short, the denoising algorithms and the statistical denoising algorithms that we use to employ before the clustering stage has actually enabled us to identify or to characterize these microbial communities at an increased resolution in the recent years. And this has been actively employed in methods like KIME to over the existing OTU based methods in data2 pipeline and etc., or in traditional metagenomics pipeline, etc. So once we have identified these clusters and their representative sequences are actually estimated, and what do? We actually take these sequences, OTU or ASV sequences, and annotate them by mapping onto reference database. So this reference database here we use is the SILVA database, which contains taxonomic information for different domains of bacteria, archaea, and eukarya, and their sequences of this 16S rRNA gene tabulated, and they are stored in this repository. By comparing the sequences that we have identified in these ASVs and to their respective rRNA sequences and their hypervariable regions in the SILVA database, one can associate these ASVs or OTPs to their specific taxonomic ranks and their specific genus and classes of these bacterial species or different microbial species. So based on this mapping of specific regions that are amplified, as we already spoke about, from the 16S rRNA gene in the bacterial ribosome, these ASVs or OTUs can be assigned to be belonging to specific taxonomic class of bacteria, and this will help us quantify the proportionate level or abundance level of these bacteria in this community. So each microbial community can be studied by comparing with each other, and their abundance level can be compared, and the abundance level can be related to abundance level from the similar communities can be related to different conditions or different individuals, and generally we can create or we can estimate a generic and a normal microbial abundance level for healthy individuals. That is what is the aim of Human Microbiome Project, as we already spoke about. So this analysis, right, includes, for example, generally includes, unlike other omics data analysis, this analysis includes in-depth understanding of the data preparation, understanding what are the reference databases that are actually used for the mapping, and specific analytical approaches to answer specific question based on observation and evidence from our results, right? Importantly, we will also discuss how the sequencing data represents only one type of microbial omics, with many more becoming increasingly available. For example, yeah. In addition to microbiome or bacterial species, we can also explore the abundance levels of the virus present in these microbial communities from the virome database, and we can also explore the presence of different classes and different species of fungi present in any sample. Such studies are helpful. For example, the virome studies are helpful in understanding the pathogenicity of the existing virome in our microbial community. And fungi database or fungi identification is very, very beneficial in understanding the environmental effects on understanding the environmental condition for agriculture and agri-genomics in general. So generally, the questions about the role of microbiome members and their communities are now being answered primarily using these next generation sequencing genomic data. And sometimes this can also be supplemented and complemented with mass spectroscopy approaches to study proteomics and metabolomics. So what is generally understood in last 1 or 2 years of microbial metagenomic studies, the next phase of microbiome research lies in the ability to integrate multi-omics data sets that will combine this next generation metagenomics data set with the mass spec or structural data or metabolomics data to relate the functional metagenomics with their phenotypical informations of the metabolites that are generally used for quorum sensing or generally used for the well-being of the tissues or the environment that these microbial communities are sampled from. So in addition to this, we also see that we spoke about the gut and skin axis. So the relationship between the microbial communities that one observes in the gut level and their effect on different health conditions like chronic health diseases at gut liver axis and neurodegenerative diseases at gut brain axis are being actively explored at different levels. So to study them, such integrated approaches are necessary to understand not only the association of these abundance levels of these microbial communities to the different disease state and their influence on the metabolites that are being actively enriched and suppressed and are broken down and are being saved, are also have to be integrated in this analysis to understand their disease association and healthy general state of healthiness association also, right? So we have so far discussed a lot about the history of microbiology and the emergence of the sequencing technology and how these data has been used and can be used to study samples that that cannot be understood by traditional methods like staining or culture-based methods. So let me actually point you to some of the additional resources that you can make use from the Learn Omics Logic platform to study these topics in greater detail. Right? These resources also include practical analysis methods and tools that you will get tremendous experience with using a variety of data sets that we will also discuss in a short while. Right? In the following sessions in this program, we will look at some of these resources and lessons from these resources. We will look at example data sets. We will run the analysis together, and we will get an understanding of the tools, methods, and examples that are generally used to study microbial community and their association with different diseases and different health conditions in general. So let me give a couple of examples. So this Srigauri also pointed you to how to access these courses and associated resources from the Learn platform. So the materials in these resources are generally a course that includes metagenomics lessons and many different example projects that includes data from different model systems. For example, this data actually takes model system. This data actually contains samples from human gut microbiome, and this data contains samples from model systems such as mouse, and this data contains, or this data integrates the observation that is made in one publication and their relationship to understand the microbial community and their symbiosis, dysbiosis effects on Rett syndrome, a neurodegenerative disease affecting kids or children. So this we already know, and these courses are designed to provide you with proper explanation on the objectives, on tutorials with examples of practical applications, and etc. And the reviews of those materials have already been done by Sri Gauri earlier, so I'm not going to go over this. So let's discuss a little bit about some of the practical aspects of these approaches and what we will discuss in detail in this program. So the structures of the coursework and other details you already have been briefed, and you can also get an idea by going through the associated resources. So what do we have in general? So we understand, or we structure the course in such a way that you understand the basic concepts of applying metagenomics data and analysis for this metagenomics data at different levels, right? At their functional level, at their downstream analysis level, and at their specific downstream analysis level in R or Python's environment to specifically address the questions and hypotheses that one can generate for the data or for the sample that we have in general. So this also Sri Gauri went through this. So this is example of what you can get and what you can access from the tBioInfo server, where we have resources like demo pipelines, which gives you access to how to set up this analysis pipeline. And once you have completed setting up this demo pipeline, that will give you access to the output files that are generated through these analysis methods. You are free to download them and explore them using methods like Excel, and the pictures and the interactive outputs can be explored for their association and for understanding the patterns in the data, etc. So you are free to go through only in the metagenomics level, but also at the different omics data level. So the visual interface of tBioInfo platform can be used to study metagenomics approach at the whole genome level, and at the data to amplicon sequencing and also at kind to analysis methods, which uses ASV based bacterial taxonomic identification in comparison to OTU based identification in data, right? And behind many of these methods, we have this analysis, or we also have important lessons on downstream analysis that can be performed using the data that you can download from the metagenomics pipeline, and subjecting that to downstream analysis using popular coding languages such as R and Python. We have explained this in tutorials, and you will have access to those codes and how to understand these codes also. And these coding tutorials actually start at the beginner level. So if you are not familiar with this coding platform, you do not have to fret. You can either base your understanding and analysis only on the server-based methods and code-free analysis methods. But if you want to explore more into the coding environment of understanding the downstream analysis performed on the metagenomics data, our tutorials and our lessons include specific code blocks for data importing, for preparation and pre-processing of the data for the analysis, and for standard visualization to understand the patterns in the data, and processing of the data and performing this analysis of the data to relate the patterns observed in the data to the phenotypical condition, not to the biological conditions of the samples generally. So yeah, more specific packages and these publication ready outputs can be accessed from these code-based tutorials and also from these output and also from these pipeline outputs that we will cover more in the advanced lessons. And yeah, these are a couple of examples of what you can generate from these metagenomics pipeline. And so for before going or before briefing some of the practical examples, some of the projects that we will discuss during the course of this program, let me stop here and ask if you have any queries until now. If not, just let's move forward and discuss what can be studied, and that will give you generally an idea about the potential of metagenomics data and to where it can be approached or where it can be applied to. We have discussed a lot already, and we will gain some more information with some specific publication examples and practical examples now. Okay. So there are many applications. This already we spoke about, including skin condition, including food sensitivity of the drug of the gut, including autoimmune disorders and cancer, antibiotic resistance, drug resistance, and even sometimes ecological studies that can be performed based on the metagenomics application or a data application. So in this specific program, you will learn to find the data. You will learn to analyze the data from repositories like Human Microbiome Project and from repositories like NCBI, and from generally obtaining data sets that include data sets from American Gut Project and etc. that we will also talk about. Right? We will also look more into some of the control studies that can be performed on animal models, as we discussed, where the environment and the environment in which these microbiome are actually harbored can be customized and can be specifically altered to understand the potential and to understand the effect of this microbiome to their phenotypical characteristics and to their behavioral aspect in this specific example that we will discuss in the program generally. So in general, this microbiome research seems to be involved in many more important areas of microbiome or biomedical research. For example, they are actively currently used in screening for diagnostic signatures of chronic health diseases, and they are also used to understand, for example, in precision medicine and in their response to treatment efficacies and etc. And that are also explored, and the therapeutic considerations for microbiome transplantation studies are also performed, and their relationship to conditions such as Alzheimer's and cancer also actively explored. So these are all some potentials of these metagenomics and microbiome based research. And I have a question before moving on. Let's see. Stella asks, can I use 16S full sequences, long read sequences to analyze bacterial community as well as functional analysis, metabolic pathways? Okay, when the moment that you say 16S sequencing, you are restricting yourself to 16S rRNA gene or the hypervariable regions present in the 16S rRNA gene. So when you have such limited sequencing potential, you cannot perform pathway analysis or functional analysis. We will not get or gain any information on the gene or any other pathway potential of the bacteria or of the microbes that are involved. If you want to study genetic or gene level functional analysis of the genes from these different microbes, you have to perform whole genomics or whole metagenomics or whole metatranscriptomic sequencing, and then relate that or identify their sequences to specific ASVs or OTUs in this case, and then you can perform or identify genes from these different microbial we cannot say partners, we can say microbial samples and microbial individuals, and relate that to pathway analysis, related to functional analysis, related to metatranscriptomics analysis and metabolite analysis, and etc. So we have a technical question on, do primers used for amplification will introduce biases? We will actually get rid of these amplification based adapters and primers at the pre-processing stage. That at the pre-processing stage, one of the important steps that we do is to get rid of the adapter sequences and get rid of the primers that is used in the library generation method. So let's move on. Let's see, what do we have some more. So this program is mainly designed to highlight the role of big omics data analysis that is that can be used to understand not only metagenomics but at different applications of omics, big data, bioinformatics, analysis on cheminformatics, and different therapeutics and diagnostics approaches, and how analysis on this structured data in general is transforming the biological research in general, right? You can explore the other areas that is present in the Omics Logic platform to make full use of the platform. So this is to highlight the importance that the microbiome is not only relevant in biomedicine or biomedical research, but it is an important part in understanding biotechnology of soil microbiome and agriculture or agrigenomics in general, right? Metagenomic studies on agricultural applications are actually developing, and they are showing promises, and many have already started to. Many research laboratories and industries have already started using this approach to improve the yield or change the conditions to improve resistance of crops to specific pathogenic attack in general, etc. And this, in addition to that, in the current scenario, they can be used to understand the microbial diversity and their relationship to hygiene, antibiotics, antibiotic resistance, to general risk factors of urban living, including pollutants and etc., and how they have an effect on gut microbiome, and how these can be related to, for example, in contracting or in adverse effects of in term when one contracts COVID-19 disease in general. So some of the publications have already started exploring lung microbiome and their involvement in the adverse effects of this COVID-19 disease. So if you are interested in that, you can check a couple of publications in there, right, which relates to the lung microbiome, not the gut microbiome, lung microbiome, and the disease severity for similar viral load on different patients with a different lung microbiome abundance levels. Right? So at the end of this program, we aim that you should be able to find, analyze, and interpret metagenomics data on your own. And if you have any queries so far, please put that in the chat box as we mentioned. So you can also get in touch with us using different communication methods that Sri Gauri and Spurs is actually explained to you in how to reach us. So before the next session, I urge all of you to start going through the course Metagenomics and a couple of introductory lessons on the topics that we already discussed today, and that will help you with digesting a lot of information that we discussed on today and prepare you for the sessions, that is for the next session, which is going to be on understanding the sequencing technologies for metagenomics data analysis. So with that, I conclude today's session, or my portion of the today's session, and hand over the podium to Sri Gauri back or Sparsh. I'm afraid I do not know.
Yes,
thank you so much, sir. Yeah, thank you, Sri Gauri. Can I take it ahead? Okay, thank you. Thank you so much, sir, for such a wonderful session. I will now take up the registration part and help all the participants get started with the program. So this is I can share my screen. All right. So this is the program page for Metagenomic Data Analysis program. And today we had the first session, and a lot of participants had some questions regarding how this online program will be structured. So we thought of inviting all the interested participants to the very first session to give them an idea of how it would be conducted. And that is why the session was free for all. If you wish to become a part of the other sessions that we have scheduled, the schedule can be seen on the program page itself. If you scroll down, you can see that all the sessions with their dates and what topics would be covered on the dates, on these dates, along with the associated resources I mentioned on the program page. So if you wish to become a part of the other sessions, you can proceed ahead with your registration now so that it would be easy for us to share with you the links to join the sessions and also give you access to the resources to get started and join our community of bioinformatics researchers. So this is the session schedule that you can follow along. We have the dates mentioned here, the topics that would be covered for each of these sessions. And along with each topic, you have some associated resources that you have to follow along to complete each session. These resources are lessons of the metagenomics coursework on the portal, which includes the concepts, session videos, and then you also have to attend some quizzes and assignments to make sure that you have got the understanding correctly and submit your assignments on our support account. Also, once the session is completed, let's say you were not able to join the live session or you are not available at that time, or you want to revisit the session, you'll find the recording of that particular session on the program page itself. So today we had the first session. Today was an introductory session where the mentor introduced you to the field metagenomics and 16S metagenomic sequencing, and also gave you an overview of the entire course and the tools and resources for this program. So after this video has been processed, I'm going to upload it on our program page, and you can access the video of the program page of the session on the program page. Only the registrants of the program page will be able to have the access. So if you are interested to become a part of this program, please make sure that you register before we close the registrations. Thank you so much, sir, for answering Banu's question. I was just in the chat looking at the questions. All right. So this was about session schedule, how you will get the access to the recordings, the resources. Now let's jump on to the registration part. I request the participants to please mute themselves. All right. So how do you register yourself? Now here we have 3 different registration days, or I'll say 3 different subscription types. The first one is where you enroll for 45 days and get the access to the resources for 45 days. And here you will get the access to the session recordings, the same sessions as we had today, and also the coursework on the Omics Logic Learn portal. The participants who enroll or take up the 45 days of subscription will get an educational license where they can run their example pipelines with the data sets given in the coursework and practice along to see how the pipelines work. All right. And then you have the second subscription that is for 60 days. So once the program is completed, once the training with all the sessions is completed, the participants with 60-day subscription will get some extra time to meet the mentor one-on-one, have these one-on-one sessions with the mentor, and work on a research proposal to get started with a research project. So they learn, get trained, and learn how to work with metagenomic data and carry out the analysis. And they finally apply the knowledge that they gain to answer a research question in hand. So that is about the second type of subscription level, a 60-day subscription. Then you have the third subscription that is for 90 days. Here, the participants will get to work on a research project and get it published. The outcome of the 90 days subscription will be a publication where the participants, first of all, complete the training program, and then they submit their research proposal, and then they start working on their research project. And if everything falls in place, if they're able to reach the deadlines, then the outcome would be a research publication. The outcome for the 60 days is a poster presentation. That means that you could present your research work in conferences as a poster, as a research poster. And if you are from a different country and not US, you can simply click on this icon here, which shows all the different currencies. And you can click on this and see what it would be in your own currency. Now we are offering scholarships to low-income countries and also to students. So please reach out to us on communications@pine.bio and also marketing@omicslogic.com. It would be better if you reach out to Sparsh directly at marketing@omicslogic.com for any questions that you have, and also thank you so much, Shrikari, for sharing the details in the chat box. We have the WhatsApp number as well active. This is a company number, so you can reach out to Sparsh for this. She's the marketing manager, and she'll be able to help you out with your enrollment and registration. And also, if you are seeking scholarship, I'll just quickly share the scholarship form in the chat box. Please fill out the form, and we will review your application and get back to you on it. If there are any questions, please put it in the chat box so that we can pick it up and help you get started. Here is the scholarship form I have shared in the chat box. Yes, here it is. Please fill the scholarship form and we'll be able to help you get started with the program with a scholarship. That was it that we wanted to discuss today. If there are no more questions, I think we are good to end today's session. But if you have any questions that you would like to clarify over an email, then you can reach out to Sparsh on her mail ID. It is shared in the chat box. We are looking forward to meet you all for the second session. And in the second session, we will be discussing about the first pipeline and also the microbiome functional bacterial community. We'll get you all to a practical hands-on the very first pipeline, where you'll be able to classify the OTUs based on taxonomies, based on taxonomy level, to be more specific. The second session is on September 29th, and we are looking forward to meet you all for the second session. Thank you everyone for joining, and have a nice day.
Sonalika, we do have a query. All right, listener, we'll mail you the answer. All right, thank you, everyone. I hope to see you all for session 2.
Yes, I can take that question, Shri. Okay, we have a question from Lizna. Question on research project completion. Does the 90-day program require to finish a project within the period to research publication? Yes. When we have a program for 90 days for research fellowship or research internship, then you will be required to complete the project within the 90 days. And that is the usual time period for many participants like you, Lizna. So it is not a tall task. We will discuss on what level are you currently and what do we need to learn to promote you to the level of performing these analysis. And that we will design a research question for which you can perform the perform the project and work on the project and get the answers. And depending upon the promise of the project, you can work with you can decide on the research publication a little bit later. Okay, thanks.
Thank you so much, sir. So if no one has any queries, we will end today's session, and I hope to see you all for session 2. Thank you, everyone. Have a nice day.
Thank you.
Thank you, Bendik.